Okay, hello. We're going to start talking about meiosis and how it's different from mitosis. Now, meiosis is ultimately a very different kind of cell division, and it occurs because sexual reproduction is going to be the combination of characteristics. When we take a look at parents and offspring, the offspring have characteristics of both parents, and that's because we've subdivided the genes in a very specific way so that each offspring gets exactly one half of the parent's genes and a specific half. And this is going to be the source of heredity. And how those genes are split up is going to create variation. And variation and the study of that variation and the result of variation is going to be integral to how organisms change through time. So, when we talk about this process, we're talking about the movement of genes. Um, and those genes are passed on via gametes. So meiosis is going to be related to the production of gametes, sperm and egg. Now, in humans, that DNA is packaged into chromosomes. We have 46 total chromosomes in what are called somatic cells, but in um, uh, testes and ovaries, we're going to make sperm and egg with 23 chromosomes. Now, on those chromosomes, there are genes, and each gene's specific location is referred to as its locus. Now, this is going to actually be different. This terminology is going to be different than what we're going to present in Mendel, and that's largely because Mendel doesn't know anything about genes and chromosomes and DNA, but he gets to the same point, and that's kind of the genius of Mendel when we get to Mendel and talking about that. Now, um, meiosis is going to be involved with sexual reproduction. And let's distinguish that from asexual reproduction. Asexual reproduction is where we're getting offspring that are identical copies of the parents. You might have heard the term clone or cloning. Sexual reproduction, on the other hand, is going to be where we're going to have two parents that are contributing to the genes of the offspring. Now, asexual reproduction occurs. It actually occurs fairly widely. It can occur in, say, bacteria when we talk about that sort of binary fission, but it also occurs in eukaryotic organisms. And that's actually something that occurs, but not universally. And we'll talk about that when we get to Mendel and sexual reproduction. We'll talk a little bit about why sexual reproduction is important. But recognize that asexual reproduction does occur in multicellular organisms. So, for instance, in a hydra, you get budding, where one parent hydra buds off and makes baby hydra. In a lot of plants, you can have asexual reproduction by some sort of a splitting. So, for instance, redwoods, um, the parent redwood may fall, but then out of that rootstock, we might get other organisms, or you may have starts, uh, uh, trees popping up from the roots somewhere else, but they're genetically identical. Now, when we talk about sexual reproduction, what we're going to be talking about is the movement of chromosomes. And we saw the movement of chromosomes in mitosis. But let's talk about those chromosomes a little bit more. Okay, here we have an organism that has a diploid or 2N paired set of chromosomes of 6, which is actually going to be two sets of 3 chromosomes. And in this case, we're going to have 6 total chromosomes, but 2 paired chromosomes, a red and a blue one from the father, one from the mother. And at this point, they've actually been duplicated. If we take that to, say, a human, this is what we call a karyotype. And we're going to have 23 total chromosomes. So 23 pairs, that's 46. And so we've got a pair of chromosome 1, one of which came from your father, one of which came from your mother, a pair of chromosome 2, one of which came from your mother, one of which came from your father, etc., etc. And the only difference with those chromosomes is that we've got, in this case in humans, 22 what are known as somatic chromosomes, or the term is autosomes. These are going to be found in all cells, and they are identical. Well, not identical because one comes from mom, one comes from dad, but there's no differences between, say, chromosome 1 from mom and chromosome 2 from dad, chromosome 1 from dad, in what genes are present. On the other hand, there's a 23rd chromosome in humans, and those are the sex chromosomes, XY or XX. And that's going to be the chromosomes that are going to determine, at least in humans, your genetic sex. It is not actually, well, there's some connections, but recognize that what your chromosomes are is not necessarily what you are. And that's because there are some connections here between genotype and phenotype. And we'll talk about that more when we get to Mendel and how heredity works. Now, thus, a human life cycle. Okay? This is sort of the standard animal model where you have a diploid multicellular adult and they produce haploid gametes, and those gametes combine to fertilize and create a zygote. Most of your life cycle is going to be diploid, and a little of it is going to be haploid. But recognize that there is actually variation in life cycles. There are different ways to play the game. In fact, there are three basic patterns that we see in eukaryotes, or at least multicellular eukaryotes. We've got the animal model on the left. So a diploid adult produces haploid gametes, fertilization, and then a zygote that develops into a diploid adult. On the right side, all the way over on the right side, we have sort of the fungi model and some protists, some other organisms, where the organism is haploid. So we have a haploid unicellular or multicellular organism, and it's going to produce gametes by mitosis. It has a single set of chromosomes, whatever that number n is, and it's going to make gametes by mitosis, just copying. Those gametes get together in fertilization and we get a 2N zygote, a diploid zygote. But it then undergoes meiosis and produces generally haploid spores, or generally spores which are haploid. Okay? So it's kind of the reverse of what we see in animals. Plants, on the other hand, go back and forth. In plants, and that's the center diagram, we have what's called alternation of generations, where we go from a diploid multicellular organism that undergoes meiosis to produce spores, and those spores go off and grow into a haploid multicellular organism, which produces gametes that come together to form that diploid multicellular organism. In plants, we refer to a sporophyte. That's the diploid. Sporo, spore-producing, phyte, plant, sporophyte. And then we have the haploid gametophyte, gamete-producing. So there are different ways to play the game. You can play it in a multitude of different ways. Now, when we do meiosis, and this is the sort of the unique aspect of any sort of sexual reproduction, meiosis is what we refer to as reduction division. And so what it's going to do is it is going to take a paired set of chromosomes and produce offspring that have only one set of chromosomes, one of each pair. So in humans, we would go from the diploid 46 to the haploid 23. Okay? So that's why we call it reduction division, because we are having the available chromosomes. And because we are having the chromosomes, we are going to do it in 2 stages. So that's one of the big differences between mitosis and meiosis. Meiosis is going to have 2 stages, meiosis 1 and meiosis 2. And so 2 consecutive cell divisions. And we are going to fundamentally split the chromosomes in meiosis 1 in a different way. So we will end up, after the end of this process, with 4 daughter cells. Mitosis, we had 2 daughter cells. Meiosis, we're going to have 4 daughter cells. And those daughter cells are going to be different from each other. So in mitosis, we produced 2 identical daughter cells that are identical to the parent. In meiosis, we're going to produce 4 non-identical daughter cells, each with half the number of chromosomes as the parent cell, and not the same as each other. Okay. Now, same sort of process as we saw in mitosis. We're going to duplicate the DNA, so we're going to have S interphase before we start the whole process. But in meiosis 1, we're going to do something different. Okay? What we're going to do is we are going to separate what are known as homologous chromosomes. And then in meiosis 2, we're going to separate the sister chromatids. So we're going to start with duplicated DNA. Meiosis 1, we're going to separate the pairs. So each of the daughter cells at the end of meiosis 1 is going to have either dad's or mom's chromosome. And then in meiosis 2, we're going to separate those 2 sister chromatids that are present in that X-shaped chromosome to produce the 4 4 daughter cells. So, interphase, we're going to duplicate the DNA. Meiosis 1, we're going to separate the homologous chromosomes. In meiosis 2, we're going to separate the sister chromatids. So, how are we going to do that? Well, what we're going to do is we're going to line up the homologous chromosomes in a very different way in prophase 1. Okay? Instead of treating each of the chromosomes, each of the X-shaped structures individually, we are going to create what is known as a tetrad. Tetra, 4. So this is going to have 4 sister chromatids. We're basically going to bind the 2 homologous chromosomes together, and then we're going to split those up. So prophase 1, we form these tetrads, these structures of paired homologous chromosomes. And so we're going to line those paired homologous chromosomes up along the metaphase plate. And then during anaphase, we're going to split the paired homologous chromosomes. And so if you take a look at anaphase 1 here, it doesn't look like anaphase in mitosis. And then we're going to do cytokinesis, and we're then going to have what is known as interphase. We're going to do no DNA duplication at this point, and then we're going to go into meiosis 2. Okay, so meiosis 1, very different beastie from mitosis. And that's because in prophase 1, we're going to organize, we're going to arrange the chromosomes in a different way. By the way, this is going to take a long time. It's very complicated. And so prophase 1 ends up being much longer than prophase of mitosis. Now, we now have 2 daughter cells, each with, at this point, half the number of chromosomes that the parent had. Okay, if this were a human cell, we'd end up with two daughter cells that have 23 chromosomes, but those chromosomes are still duplicated. They're still X-shaped structures. Okay? So in meiosis 2, what we're going to do is we're going to separate those X-shaped structures. Meiosis 2 looks pretty much like mitosis, except that the two cells that we produce are going to be non-identical. If you take a look at the red and the blue, you'll notice that we're not getting chromosomes that are completely red or completely blue. And this is because in prophase 1, back in meiosis 1, we did something called crossing over. We'll talk about that in just a sec. But this is similar to mitosis. Basically the same process. We're splitting up those X-shaped chromosomes, but we're doing it on a haploid cell because we've split those chromosomes. We now have just 23 chromosomes if we're looking at a human. And by the way, don't get caught up in 23 and 46 as unique numbers. Pea plants, for instance, have 128, something like that. Fruit flies have 4. N is different for different organisms. It just happens to be 23 and 46 for humans. So you can watch this process.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
one. Okay? This is the fundamental place where meiosis becomes different. Okay? Because during prophase 1 of meiosis, what we're going to do is we are going to have those paired homologous chromosomes line up together. Okay? This process is called synapsis, and essentially what we get are proteins that bind the chromosomes together. These proteins are actually present in mitosis, but they only bind the sister chromatids to themselves so that they separate. Whereas in prophase 1, these proteins called cohesins are going to bind the two sister homologous chromosomes together. Okay, so here's a diagram of crossing over. Okay, so we have one chromosome from dad, one chromosome from mom. This is from your dad or your mom, and you are passing those on to your offspring. So if we're looking at it in the perspective of the child, this would be each parent is passing on either grandpa or grandma's from the two sides genes with some variation because of a process called crossing over. You see, these two homologous chromosomes are set up the same way. Essentially, you've got two bookshelves, and all the books on the two bookshelves are arranged in the same way. The only difference is that the book on the top shelf, 3 from the end in Dad's bookshelf, might have a slightly different version than the same book in the same position as Mom's bookshelf. These are individual genes, and the different book is the different version of the gene that you might get from one or your other parent. Okay, so what this means is that these two homologous chromosomes are very, very similar to each other, and there are places where they're so similar that they can swap. These swapping events are called crossovers. And what they do is they form chiasmata. The Greek letter X is a chi, and so chiasmata, crossing over. Okay? Now, what this does is it means that you can pass down to your offspring not dad's chromosome or mom's chromosome, but you can pass down to your offspring a chromosome that is part mom and part dad. It's like shelves of the bookcase are being swapped. So you get mostly mom's bookcase, but you get shelf 4 from dad. And vice versa, you'd get mostly dad's bookcase, but you get shelf 4 from mom because they've swapped. Okay? This crossing over is actually very important both for chromosomal continuity and also for creating variation. So, what does this mean? Okay, well, mitosis and meiosis are going to be different. Mitosis is going to produce offspring that are identical. That is, that the number of chromosomes are the same and the genes are exactly the same. There's no variation. Meiosis, on the other hand, halves the number of chromosomes. We go from a diploid cell to a haploid cell. And because of how they're passed on, each one of those cells is genetically different from each other and from the parent cell. Okay? And that happens because of 3 things that are unique to meiosis. That synapsis of chromosomes during prophase 1 is going to allow for crossing over, for mixing and matching of the chromosomes. Okay? And then when we line up the homologous pairs at the metaphase plate, we can get either dad's or mom's chromosome going to one end. Okay? So if you get dad's chromosome 1, you might get dad's chromosome 2, but there's an equally likely chance you get mom's chromosome 1, mom's chromosome 2. What happens to chromosome 1 is independent, and it's actually called independent assortment of what happens to chromosome 2. And then when we separate those homologs during anaphase, that's what's going to create the difference and the synapsis. So, just as a diagram of this process, we have mitosis on the left, meiosis on the right. And in meiosis 1, we are going to separate the homologous chromosomes. So at the end of meiosis 1, we're going to have a haploid cell, but one that has non-identical chromosomes because of any potential crossing over. And then we take meiosis 2 and we separate those sister chromatids to produce 4 total individual cells, and they are not identical to each other. Okay? And so what this does, and sort of the whole point of sex, is to produce genetic variation. Okay? Genetic variation occurs because of mutations. So say you have a mutation in the cells that are producing your eggs or sperm, well, those mutations, those changes would be passed down to your offspring. And so those are going to be random changes to the DNA. But in addition, we're also going to have reshuffling. That crossing over is going to allow us to reshuffle, mix and match the alleles, the different parts of those chromosomes during meiosis, and therefore also producing or magnifying the genetic variation. And so we're going to end up with several things that sexual reproduction is going to do and going to create that genetic variation that we see occurring through sexual reproduction. Independent assortment of chromosomes, crossing over, and random fertilization. So this is going to create genetic variation. Okay, so let's start with independent assortment of chromosomes. Okay, what it means is that during meiosis 1, during metaphase specifically, how those homologous chromosomes are oriented on the metaphase plate is going to be random. And so that means there is independent assortment. Whether or not you get chromosome 1 from dad or mom does in no way affect whether or not you get chromosome 2 from dad or mom. Okay? So, what's the net effect of that? Well, that means that each chromosome is a coin flip, whether you're going to get dads or moms. In humans, there are 23 total pairs of chromosomes, so that means that there is 2 to the 23rd different ways to arrange the chromosomes. That's like 8.3 million different ways. Plug it into a calculator. It's 8.31 something or other. Yeah. Okay. So independent assortment is going to alter how the chromosomes are passed down. So you can, just with 2 sets of chromosomes, okay, you can get 2 to the 2 different ways it gets arranged. You could get one copy of dads and one copy of dads. You could get one copy of moms and one copy of moms. You could get chromosome 1 from dad and chromosome 2 from mom, or chromosome 2 from dad and chromosome 1 from mom. Okay, so 2 to the 2, 4, 2 to the 23, 8.3. If you guys have siblings, this is one of the reasons why you probably don't look completely like your siblings. You're going to share, on average, half of your genes, but on average only half. Okay? Now, on top of that 8.3 million different ways we can arrange the chromosomes, well, we're not even counting how those chromosomes have been mixed and matched by crossing over. So 8.3 plus or times all the amount of crossing over that occurs. In humans, that's an average of about 3 crossing over events. So, yeah, the numbers get huge. Just a diagram of that process. And you notice that at the bottom, the daughter cells, you can get chromosomes that are arranged in very different ways. Another animation to look at. And okay, so we've got independent assortment, we've got crossing over. Well, we've also got random fertilization. Okay? That is, for instance, it's whichever egg is available and whichever sperm gets there first and combines with it. So if we take 8.3 million times 8.3 million different options, literally a human, the chances of two siblings being identical, except for identical twins, and that's a sort of a separate issue, is 1 in something like 70 trillion. And that's not even counting crossing over. So each zygote is going to be different, almost certainly. The one exception is identical twins, and that occurs because identical twins are actually the same fertilization event, but then the zygote splitting into two independent organisms. Now, we've gone to all of this complicated process. Meiosis takes a whole lot more work than mitosis. It is much more complicated. It takes a whole bunch of more bits and pieces. Sex, human sex, well, it's really about getting sperm to egg. Everything else is bells and whistles. But, we spend a lot of time on those bells and whistles. Why? Why does sexual reproduction occur? Because it seems like a very inefficient and error prone, and it is in fact much more error prone than mitosis process. So why does it occur? Well, it occurs because it creates variation. And variation is necessary for natural selection. And natural selection is the driver of success in organisms. Organisms that are going to be asexual are going to have much less variation, and therefore, if conditions change, or when conditions change, they're going to be less able to respond to those changes. And this is one of the reasons why most complex organisms, most eukaryotes, are going to have some sort of sexual life cycle, because it is the driver, it is the creator of the variation that is necessary for natural selection. And so that's why we do sex. So that's it. Next step is to talk about um, the process of Mendelian inheritance, sort of what are the results of sexual reproduction? Can we follow these patterns, and what are those patterns? How do they work in organisms? So next step, Mendelian um, uh, Mendelian processes, understanding the heredity of how organisms work in this animation. So, and I'd recommend you take a look at it because it kind of walks you through. But let's talk about prophase